Maybe just very brief, yeah. So maybe we will just start from here. And I hope everybody can see my screen and hear me.
Yes.
Yes, okay, great. So, okay, we will, so what we were discussing is once the progenitor proliferation, very initial steps of progenitor proliferation, like the very symmetric divisions which happen at embryonic day 10.5 of the neural epithelial cells, which then go on to make this radial glia, which are also called the apical radial glia. They're also called the ventricular zone progenitors. And these have the apical end and the basal processes which go and touch the peel surface. And these then give rise to all the 6 layers in the neocortex. And they all do so by first initially by direct neurogenesis, giving rise to neurons, which forms a very nascent initial structure called the preplate. And I'm not going into the details of all of that. But once the layers formation starts, then it starts making intermediate progenitors, which are also basal progenitors, in the sense that these progenitors, there are differences between these, not just in terms of markers, but in terms of their division as well. If you remember the video which I showed to you, that the apical undergo vertical divisions, whereas the basal undergo horizontal divisions. And they have very restrictive potential in that they may undergo one more round of division, or they can directly give rise to neurons. So every intermediate progenitor gives rise to 2 neurons, which migrate and they go and populate the cortical plate. In this case, this is a formation of layer. This is then followed by the same progenitors dividing and give rise, making basal intermediate progenitors, which divide and give rise to the layer 5, which migrates past layer 6 and sits on top. And in between, you also have this formation of outer radial glia, which are also called the OSPZ progenitors, which are also called the basal radial glia, which have more division potential than the basal intermediate progenitor. And they are found very less in mouse, and I'll be coming to that in a bit. They also contribute towards the neurogenesis and give rise to the different the layers in the developing cortex. And towards the very end, that's postnatally. So what happens in the cortical development is that the period, the onset of neurogenesis is embryonic day 11.5. This is mouse brain development. Until about, say, 17.5, 16.5, 17.5, the neurogenesis happens in the cortex. And the peak of neurogenesis is between 12.5 to 16.5. So at around 16.5 onwards, the neurogenesis starts to taper off and the gliogenesis takes off. And the gliogenesis takes off at 17, 18.5, just when the embryo is being, the pups are being delivered, and postnatally, the glia generation picks up. So in mammals, the neurogenesis and the gliogenesis is temporarily regulated in that first neurons come and then glia come. And mechanisms of which I'll not be going, but perhaps some of you, if you're interested, you can read some very nice reviews which talk about what mechanisms regulate this neuron glia sulfate switch. So shown here is postnatally how this very same radial glia becomes a progenitor, astroglial progenitor, which will then give rise to glia, which go and this is the white matter, and which then give rise to all the cortical astrocytes in the cortex, right? So I told you in my last class about this, how these progenitors generate neurons in an inside-out fashion. We also talked about the different neurons. I'll not be going, this is just a image, and also the importance of renin, because in the absence of renin, how the cortex gets inverted. And okay, and this is where we had perhaps stopped, or we were discussing that how this entire temporal specification is regulated by the expression of transcription factors in the progenitors, in the SPZ progenitors, which are the intermediate progenitors. And then some of the new transcription factor machinery or combinations come up in immature post-mitotic neurons. and followed by there are more in the mature postmitotic neurons as well. And also, I mean, I talked about my own work in this, but I'll skip all of this. But I think what I want now to tell you is all these studies using bulk, using genetic, and for instance, if one were to understand that, sorry, let's focus here. So the question still lingered is, if we were supposed to look at the development from, say, embryonic day 12.5 to 15.5, what is the molecular signature of these progenitors as they go from 12.5 to 15.5? So that is one temporal axis. The other axis is this radial axis, that the axis going from the ventricular zone to the subventricular zone to the neuron subplate and the neurons and to the marginal zone on top. So as you go, and clearly defined are three zones. The first zone, which consists of both the intermediate progenitors and just the newly postmitotic neurons, and of course all the postmitotic neurons in the developing cortex. So what are the signatures, or what are the sort of, what is the hallmark of such cells across time, the progenitors across time, and the cells across the radial thing? So single cell RNA-seq studies have now sort of changed or sort of revolutionized the field in terms of giving us a snapshot of how a cell would change in time and also across the radial axis. And this work has been done from Denis Gébaudon's lab, where I'll first describe how they have tried to understand this. So let's just skim over all of this. Yes. So the paper is in Science, and it came in 2019, and precisely talks about what is the temporal patterning of the apical progenitors and their daughter neurons in the developing neocortex. So as you can see from the title itself, it's across two axes, the time axis and the radial axis, as the progenitors go on to form neurons. So here their entire work is summarized in this. I'll first just give you a snapshot of what they found, and then perhaps walk you through some key images which will give you an idea of what's happening. So this is the embryonic brain as it looks at 12.5, and as you can see very clearly that by 15.5 that the embryo has grown, the ventricle has shrunken because the embryo is, the brain is growing, and the whatever region is marked here is what they're looking at during from M12 to E50. So one of the first things they observed from the apical radial glia as they go from E12 to E15 is the absolute change in the sort of expression of not the change, but the coming about of newer sort of gene molecular signatures, which suggested that as the progenitor goes from E12 brain to a E15 brain, you start to get some of these expression of genes which now can talk to the environment. So for instance, at E12, the genes which were mostly a large bulk of them, a big subset which came as a group, was chromatin organization and cell cycle control. And you would highly appreciate now that you yourself understood how the sort of cortical development progresses, that at E12, the division, what is really predominant is proliferative divisions, or divisions which are important to control the number of progenitors, the number of apical progenitors, the number of basal progenitors, and that's why you have a lot of these coming up in these progenitors. And as time progresses, you have expression of membrane receptors, or the ones which control membrane potential, thereby suggesting that, because by E15, the cortex starts to get inputs from the environment. For instance, inputs from, the growing axons from other places, or inputs from the thalamus or things like that. So it is that there could likely be an influence from the environment, and there where you have a concomitant expression of membrane receptors and control of membrane potential. Now, if you look at this axis on in the radial side, so one thing which they observed was there is a conserved differentiation program, which is, so whatever the status of the progenitor is, there is a conserved differentiation program. For instance, what I mean by the conserved differentiation program is there is a mechanism by which you generate the deep layers, and the progenitor then transfers this mechanism to the neuron, and then you enact out the conserved differentiation program. And as time progresses, initially it's mostly subtype specification, followed by you start expressing genes which are important for migration, axonogenesis, followed by neurotransmission. If you recollect my own slide a few some time back, I told you the emerging function of the nervous system, right? So all this is talking to each other. For instance, even the expression of genes comes about starting from progenitor proliferation, as you can see here, cell cycle control, followed by subtype specification, followed by migration, axonogenesis, and neurotransmission. And that's how the functions in the nervous system emerge. So what they did, and I'll tell you very briefly, not going over the entire, it'll become a journal club otherwise. So very briefly what they did, and I think this is the sort of, in their paper, the thing which they did is they had a molecule called FT, and that molecule would only be taken up by the apical progenitors, and at a time when they actually come to the ventricle. These apical progenitors divide, so these apical progenitors divide at the position at the ventricular zone, right? And they undergo a vertical, either asymmetric, symmetric division. And this FT molecule can be taken up only from those, because it is injected into the ventricle, and only those dividing cells which are at the ventricular zone can take up these. So basically, you're only looking at progenitors which are dividing, and which then will give rise to a differentiated progeny. So what they did, that they did this experiment across 4 time points, embryonic day 12, 13, 14, 15. They would give the FT injection, wait for an hour, harvest those cells. So basically now all those progenitors which divided in that 1 hour time point will all be labeled. And as you can see here, if you give FT at 1 hour, you would very nicely, the ventricular zone being lit up. So which means the apical progenitors are lit up. Now, if you would harvest after 24 hours, you would get both the subventricular zone progenitors and the newly postmitotic neurons. And if you harvest after 96 hours, you will suddenly see, that all the progeny, all the FT is seen in the cortical plate. So thereby they were able to get 3 subpopulations by giving one injection of, by giving the injection of FT at 3 different time points. Sorry, giving the injection of FT and harvesting at 3 different time points. And now not just you can do a temporal sort of analysis, but you can also do a renal analysis. And this is how mostly we do the analysis in the developing cortex. Okay, so just quickly, very quickly, I think this slide is just going to summarize their entire thing, which I just talked to you about, is that they found, so this is distinct signatures in the apical progenitors as they progress. So the of the cells are going from AP to PP, going to 1-day-old neuron, going to a 4-day-old neuron. And they also, because they've done this at E15 as well, because 96 hours later you get astrocytes, you can also look at interneurons. They found that the astrocytes form a very clear subset. The architecture is signature is very, very different. So is for the interneurons, which don't come from the cortex, but come from the ventral tail encephalon. So just to, quickly show you that how the layer or the cell type marker genes change. So very early on, they all express, SOX2, which is a marker, SOX2, which is a marker for progenitors. You'd also see that at the time of astrogliogenesis or astrocytes as well, the astrocyte, the progenitors which give rise to astrocytes are also SOX2 positive. And as you go from embryonic AP to C that has from SOX2 to EOMS2, which is TVR2. Then as you become just newly post-mitotic, you start expressing depending on which layer. So if you are a deep layer neuron, which is layer 5 and 6, you will express SOX5, BCL, CTIP2, BCLL, BCL, BC11, 1B, BCL1B, which is CTIP2, and FESF2. FESF2 is a marker for layer 5, I told you about it. So this newly post-mitotic start expressing these genes. But if you were a newly post-mitotic gene, which will, and if you are if you're making the superficial layers, you start expressing very early on itself, you start expressing some of these markers, which are important superficial layers, something progresses, you start that, you know, the enrichment goes from a progenitor like to a neuron like. Okay, and here also you can see that, you know, AP early marker very clearly marks only embryonic 11.5, but the AP mid marker does not get expressed at. So when I say, so now we're talking about genes which mark from embryonic day 12.5 to 15.5. So this was the radial axis, this is the temporal axis. So if you take three markers, like one which progenitor expresses in the early ones, say at E12, and then mid one, say 13, 14, and the late one is 15. So as you can see that an AP early only marker will only be expressed at 11 or maybe say 13. But the AP mid would be expressed say 13 and 15, whereas an AP late is highly expressed in 15. So that's how they distinguish their different markers. And similarly, for a neuron, this is a post-mitotic neuron. At 15.5, you would see its expression in the cortical plate, and by because Lee has is the formation of layer 5. The layer 5 is already formed, and you see that this marker exclusively labels layer 5 neurons, which you can see postnatally. Similarly, if you see a late marker for the E15 born neuron, you would see that it comes up only postnatally, only in the superficial layer. So thereby they've now been able to sort the entire cortex across the temporal and the radial axis into different kinds of markers. And also one of the things which they did, which I'm not sure, showed one of the chromatids, the repressor complex, so which means that this chromatin architecture plays a very important role in restricting or regulating the expression of these genes. And that's why my own lab is now focused on looking at some of these chromatin modifiers, the three ones which I showed to you, and looking at how they interact with one another to do these kind of expression changes. So that's one of the arm of research which my lab is working on currently. Okay, so now we've talked about a lot of mouse cortical development. It's been a very, very important and valuable system for us to understand some of these conserved mechanisms. But as I said, coming from mouse to human, there has been this huge change or the expansion of the neocortex. And that it's important now to study what could have contributed to this neocortex. So people some years back, probably say I think maybe 10 years back, or when people started looking some of these, when people wanted to understand how is the human brain development different from the mouse brain development. They started accessing the fetal, human fetal explants, which come from aborted fetuses. So that was one of the starting point where people started looking at what could be different between the mouse and the human. So as I said, there are brains which are bigger than the human cortex, that namely of an elephant or a porpoise. But important to know that the human cerebral cortex has the highest number of neurons, and that's why the expansion is considered, as an increase in the number of neurons is more in the human cortex, right? So what I'm showing you here is there are many differences between the mouse and the human. And I think we can probably have an entire class devoted to understanding what is the difference between the mouse and the human. But I will tell you some salient features and I'll talk to you about it and how studies from human fetal brains, fetal brains, or now with the advent of iPSCs and organoid technology, what are some of the things which we have studied and we will continue to study. So one of the things, as you can immediately see from the top here, is that this is the mouse brain on the left, and this is the human brain in the right. And I've also shown you a sort of sagittal section and shown you how the expansion has just completely changed the anatomical structure of the human brain. But just not that. One immediate thing which you will see and observe is the fact that the mouse brain is smooth, or that's called lissencephalic, whereas the human brain is gyrencephalic. Because the expansion has happened too much to fit the entire brain into the skull, the brain has folded onto itself, and that you can see very clearly here. This is a cross-sectional view of the mouse brain. You can see the smooth brain, and this is the neocortex here, and this is the hippocampus here. And a similar cross-sectional view of the human brain will show you that the neocortex, and you can see because the neocortex has expanded, it has folded into itself into gyri and sulci, and that's what has led to the formation of a gyrencephalic human brain. Now, if you would look at the corresponding development in the cortex embryonically, what has really happened is one thing which jumps out to you. See, this is now, you're all familiar with this mouse brain. This is an embryo on day 13.5. This marks the VZ, this marks the SVZ. VZ consists of apical radial glia, SVZ consists of intermediate progenitors. Now, what jumps out to you at the same gestational week 14 from the human brain is this expanded outer SVZ. Now, the mouse also has outer SVZ. It does not have it across the entire brain. It only has it at certain regions, like it has more ORG, it has ORGs in the medial, and you really need to look for those ORGs in the mouse brain. But what it really tells you is that ORGs did come up in evolution in the mouse and then got expanded in the humans. So as you can see here, that the outer SVZ, which consists of the outer radial glia, have hugely expanded in the mouse, in the human, and this is highly implicated in the huge neocortical expansion which we see in the humans. Not just this, the superficial layers in the humans are increased because of the outer SVZ progenitors having increased, and that has contributed to the neocortical expansion. So this is again showing the same thing, that how say embryonic 14, where this is the OSVZ, but just look at the kind of expansion which is seized by gestational week 17. So as I said, 10 years back, Kriegstein's lab started, Arnold Kriegstein's lab started looking at this from human fetal and show you one key difference between the apical radial glia and the ORG, and that's again got to do with the division. So I'm just going to show you that slide, the video here.
And we were very lucky at UCSF to get donated tissue that we could treat much the way we treat our harvested mouse or rat tissue. And this is a slice culture where we've used a virus that selectively infects only outer subventricular zone it's highly radial, highly dynamic. And if we sort of zoom in on just some of the progenitor cells, which are shown here.
Okay, so one of the things which is when now you're looking at the ORG division, one thing you should first look, total absence distinguishes the ARG from the ORG, right? That's one thing. But now
some of the mitotic behavior. So each one of the cells shown on the left divides once in this time-lapse image. and they divide in a very peculiar way. If you look at the panel, the panel here on the right, you'll see one of these cells that you can follow as it translocates and divides. That behavior, where the nucleus moves up along the radial fiber just before division, is something very characteristic of these cells, and they hadn't been described before. In fact, these cell types hadn't been described before.
Did you see just the jump which the ORG just did? And that's called the mitotic somatic translocation, or the MST. Do you want me to play it again, or?
Yeah.
Hello?
Yes.
Sorry,
Yeah.
I couldn't.
Could you play it again?
Yeah, sure.
Which are shown here, you can see some of the mitotic behavior. So each one of the cells shown on the left divides once in this time-lapse image.
For instance, what I would suggest is, okay, yeah, focus on this cell here and it'll just divide. So it'll just show you the plane of division and all of that.
So each one of the cells
So it
shown
just
on the left
divide.
divides once in this time-lapse image. And they divide in a very peculiar way. If you, one of these cells that you can follow as
okay. See that it divides, jump, it just jumps. So in case of the ARG, what really happened was it divided and there was a slow movement of the nucleus along with the daughter cell, right? Now you would see that here, there's just, it divides and it's just take a leap. And I'll come to in a bit that how the internuclear kinetic migration, or the INM, and the MST, which is the mitotic somal translocation, the two are different.
If you look at the panel, the panel here on the right, you'll see one of these cells that you can follow as it translocates.
Did you just see jump from its position in the cortex to a top, and they just divide? Yeah.
And divides. That behavior, where the nucleus moves up along the radial fiber just before division, is something very characteristic of these cells, and they hadn't been described before. In fact, these cell types hadn't been described before. So we named them, and we named the cell the outer subventricular zone radial glia-like cell, or ORG cell, or ORG cell for short. Now they're sometimes also referred to as basal radial glia. But these outer radial glia, or ORG cells, also have this unique mitotic behavior, which we've called mitotic somal translocation, or MST for short. Now these are complicated words, so I'll refer to them as their acronyms: ORG cells and MST divisions, because these are characteristics of the cell type, which is highly enriched in the developing human brain.
Okay, so coming back. Yeah, so I wanted to show you that video just to tell you that, of the many differences, one of the first differences is the fact that there exists this expanded OSPZ, and not just the expanded OSPZ, but the ORGs, or the progenitors of the OSPZ, they are different from the apical radial glia. The markers are different, of course, but the kind of movement their nucleus shows is different. This mitotic somal translocation is very different in the sense that the internuclear INM, which they are dependent on microtubules, actin and dynin and centrioles for their movement. Turns out these mitotic somal translocation don't rely on any of these. Instead, it relies on a non-microtubule myosin, Rho and ROC NM2 pathway for this movement. Besides that, people are still trying to understand what are the differences between these kind of movements, or the between the apical radial glia and the OSPZ in these cells. So yes, clearly there is a difference between the mouse and human brain in that you have expansion. Now, taking from there, with the advent, as I was saying, with the advent of IPs and organoid, the axis kind of issue is now is much simpler. For instance, you don't only have to rely on fetal explants, but you can now start, making these organoids, which are 3D-like, which are brain loans in these dishes. And that's what people have been doing. And I'll just finish off my class with giving you a few examples or the sort of questions people are asking, and also the sort of questions my own lab would like to ask with some of these organoid cultures which have been taken from patient specific samples, right? So as I was saying that, as I was saying, iPSCs and the organoids derived from the iPSCs are not just important for, so they are very valuable not just for studying human brain development, because since it's coming from a human specific tissue, you will now, people have now started seeing that the expression of the presence of OSPZ or the ORGs in these cells. But since they come from a patient specific sample, they are highly useful to study the genetics of the disease, which is very specific to the patient, and thus can be in the future can be used for making personalized disease organoids and maybe personalized drug screening and drug treatment. Right. So I'm going to show you one such study very recently. So there are a lot of labs this organoid making. A few labs which I'll tell you are at the Sergio Pascal's lab, Madeline Lancaster, Paula Orlota, just to name a few which are coming from the top of my head. And some of these people have already started experiments or already shown how these organoids, one of the first thing one would like to ask is, yes, you made an organoid which is coming from adult tissue. Do they show the same signatures, or do they show the same molecular signatures as you would if you would look at it from a fetal explant? And that's what Sergio Pascal's lab looked at, and they published this paper very early on in 2020. They compared tissue and their organoids, and they developed two kinds of organoids as seen here. They developed the cortical spheroids and the subpallial spheroids, and the cortical spheroids give rise to your excitatory neurons, and the subpallial spheroids give rise to their inhibitory neurons. The cortical spheroids also give rise to the glia. So basically, they're making the question asked was signatures or the molecular markers or the sort of development which happens in the organoids mimics that of fetal tissue. Then one thing which you would see is that the chromatin accessibility will be very similar between the two. Chromatin accessibility is essentially how the chromatin opens and to enable by this method using a transposase. So basically, you will know what are the open chromatin regions between the different kinds of neurons or between different ages, right? And then they used this to study chromatin dynamics in human corticogenesis. They also looked at whether some of the genes already implicated in neurodevelopmental disorders, are they enriched in this? This here just simply shows that some of the markers which you had seen in the previous slides, that is PAX6, which is for marking the progenitors, SADB2, CTIP2, which mark the D plates, and these marks the glia. And these are the GABA is the marker for GABA, and GAD67 is the marker for inhibitory neurons. So some of these organoids express all of these markers, yeah, I'm sorry, I'm not showing you the data from this study, but they have basic developmental, what they saw that whatever in vivo saw to a good extent recapitulated in vitro. Though I must say that there exists study from Arnold Kriegstein's lab saying that it's not an exact, I think it's a work in progress, particularly the organoid biology, and people are working toward robust organoids, reproducible organoids, which are more and more similar to what you see from the fetal tissue. Okay. Now, another work which I'd like to highlight and show, and I'll say why organoids are important models to study human-specific differences, and this comes from the lab of Anil Kriegstein, which he published in 2019, is to show, what are the changes in the radial glia, which happens between, forget mouse, which happens between the primate species. And they found some very important key differences, particularly expression of markers or gene expressions, which was very, very unique to the radial glia in the humans, and perhaps not present in closely related primate species. And also they found a few gene expressions which were found only in the primates, but not in humans. So this work, how the ORG cells differ between, say, and what I didn't mention here is that they also looked at the study. This is an organoid study, and this is coming from a primary tissue. So anytime people look at both the primary tissue and the organoids, and they found that these expressions were seen only coming from the organoid or from the primary tissue, but not from the organoids from the primates or from the tissue from the primates. So then they also looked at the ORGs, which are and they found mTOR signal, which they could see in the primary tissue and also in the organoids, was completely organoids from primates. So which means that the mTOR signaling enrichment is human specific, and it's not just seen in the primary tissue, but it's also seen in the organoids, thereby saying that the organoids are indeed a very important system to study some of these changes. So coming to what my lab now would like to go, so my lab looks at cortical development both in health and disease. So we are focused in the mouse as a model system to look at in-depth molecular mechanisms of how chromatin modifiers interact which brain will be it'll be a knockout chromatin modifier. So these will give you tease about the molecular interactions. But when we also would like to understand what are humans is different. So we use the ADBS program to understand this with the clinically dense family, which is a neurodevelopmental disorder. They have exome sequencing, which means we'll be able, it enables us to identify putative risk variants, which could be contribute to the disease. And then we would like to do some cellular molecules match the clinical assessments. And what I'm showing you here is one such family with schizophrenia. And you can see a lot of, so this family with schizophrenia highlights stones, but not very
Bhavna? Can others hear me?
Yes.
Yeah.
Yes.
Fine.
Hello. Can you guys
Hi.
hear me?
Yes.
So I thought I was losing connection, so I logged out and logged back in, but it looks like Bhavna has frozen, is it?
Yeah.
Okay, okay. Okay, so while she's coming back online, would you like to talk about some of the things that she's discussed?
But then
So
the thing is that we couldn't really focus because there was so much break in between, and
well, no. So, for example, the first part, so where she was talking about the temporal. Wait, let me put my video on. Can you guys see me? So I'm
Yeah.
also on my phone, so I probably will not be able to. I can't see who is. Yeah. So what about she spoke about Denis Jobardon's work where she was where they're using single cell RNA sequencing and using fancy. So you'll notice this, right? So with the advent of new technologies, you can ask questions now in vertebrate systems that you weren't able to ask before in invertebrates. Which you were able to ask in invertebrates, but couldn't in vertebrates because they're so complicated. And now with the ability of the technique that she described using single cell RNA sequencing, with the ability to tag neurons specifically at specific times using that flash tag technique that she spoke of, you can begin to ask questions about whether there's a transcriptional signature that carries forward a temporal information that we learned about in invertebrate systems. So do you want to discuss some of that, because we haven't yet seen those signatures in vertebrates, right? Did people have questions about that? Oh,
Hello?
looks like there's life
Yeah.
at Bhavna's end.
Yeah, sorry, I'm back now.
Bhavna,
So where did you guys
can
lose
you
me?
hear us?
Yeah.
We're talking
Hello?
about some schizophrenia or something.
Yeah, sorry. So I was just saying that, so I think I finished. Did you guys hear me talk about the mTOR signaling enrichment in the
Hello,
ORGs?
hello.
Hello. I think I'll keep my slides. Okay, I'll keep my slides just not as share, yeah, I'll just keep it like this. I think this is better. Hello?
Yeah.
Can you hear me?
Hear you.
Can you see my
Yes.
screen?
Yes.
Okay. Did you guys hear me talk about the mTOR signaling and how is it different from, it's different in the different, it's not present, it's highly enriched in the human specific system?
We did, but it'll be better if you repeat it, because the thing is your voice was actually getting distorted throughout the presentation.
Oh,
So it
today?
was a bit hard to concentrate, yes.
Ah, why didn't you tell me before? Maybe you did.
Because everyone thought that it was something on their end. So ultimately, when you were out of the Zoom meeting, we all figured that it's something wrong with your end.
Oh, do you want me to quickly go through where or okay, I'll just quickly tell you very briefly tell you. So I walked you through the understanding of the temporal patterning of the progenitors, both from say embryonic day 12 to 15, and say across the temporal angle axis and also across the radial axis, right? So that was one of the things I told you, and I told you how the molecular signatures changed from it going from say going cell intrinsic to going cell extrinsic or extroverted. They call them introverted and extroverted. So the cell cycle, a lot of cell cycle and chromatin organization genes were here in these early progenitors, whereas a lot of them were, a major pathway, biological pathway which came up in later progenitors was membrane receptors and control of membrane potential. And then as the axis went from progenitors to post-mitotic neurons, you started seeing genes involved in migration, axonogenesis, and neurotransmission coming up, right? So that was one of the take-home messages from this study. And then I went to telling you how the OSVZ is one of the key differences in cortical development. And then and I showed you the mitotic somal translocation. I highly encourage you to look at the video from Arnold Kriegstein's lab. It's very nice. It's very, very informative, and he explains it very, very simply. So if you not got that bit from my talk today, please go and look at his video. It's on iBiology website, and it's very, very informative. So I told you that some of these studies came from fetal tissue, but then with the advent of organoids, people have now started looking at and asking the question that are organoids good for people to do these studies? And this comes from the study which I showed to you, comes from Sergio Pasca's lab. where he compared between the human fetus, fetal tissue, and also from organoids, and shown that, you know, a lot of these chromatin accessibility, because as I said, as the embryo, as the brain goes from a proliferative zone to a more differentiated zone, the chromatin accessibility changes. So he recorded precisely how this chromatin accessibility changes by looking at, say, over a developmental time point. And he saw that the chromatin dynamics was very similar between a fetal tissue to an in vitro. And this is a comparison here. And he shows that they're very, very similar in the two. And he made both cortical and or spheroids, which gives excitatory neurons, whereas spheroids, which make the inhibitory neurons, he made them separately. And these are just some of the markers which you've previously seen, like PAC6, CTIP2, SATB2, which mark the different layers. GFAP marks astrocytes, and GABA marks inhibitory neurons. Okay. So here I was telling you one more study to show that how may like, for instance, is there any human specific gene expression? And indeed there is. And this is a study from Arnold Kriegstein's lab, where they looked at fetal tissue, primary tissue. This is fetal tissue, this is from organoids, and fetal tissue from primates. And they saw that there were very specific human specific genes which were expressed in the radial glia, which were not seen in the primates. And similarly, there were at least 2 gene expressions which were seen in primates, but not in humans. And not just that, I think one of the important take home from this paper was the fact that the ORGs have mTOR signaling enrichment, which is very specific to just humans. Like this primary tissue shows mTOR enrichment, so this organoids, but not the from the primates. So suggesting that it's now important to study some of these mechanisms from either from fetal tissue, but given that, you know, how difficult it's to access those, then also to study the human brain development, specific disease mechanisms. So my own lab is now, will be utilizing the ADBS program resources, where you have clinically dense families. And I'm going to show you one example of such a dense family, where this is a family where, as you can see, the black is several members of the family have the schizophrenia and bipolar disorder, and with some few controls, with a control in the family as well. So the exome, so they're undergoing rigorous clinical sort of assessments. So whatever clinically they're being assessed, people, people like me will do cellular and molecular studies and try to compare and see whatever clinical assessments do they show a similar molecular or cellular phenotype in a dish. Because clinical neuroscience or neuroscience, where people look at clinical studies, rarely give you an understanding of what happens in the disease cellularly and molecularly. So therefore, iPSCs become a very valuable system to study those. Plus, as I said, the exomes have been sequenced to know is there any mutation or a risk variant in some of these genes which could perhaps be contributing to the disease. And from the exome sequencing, I'm showing you here one such risk variant, a putative risk variant, which is in an epigenetic modifier, because my own lab's interest is looking at chromatin modifiers. And this is SEDD6, which is an epigenetic modifier which methylates histones. And then we would like to ask the question that, you know, what happens if you take an organoid from such a patient who has a putative risk variant in SEDD6, how is the chromatin accessibility changing, or how neurodevelopment is altered? And I've shown you some of the steps of neurodevelopment, going from progenitor proliferation to formation of neurons to dendritogenesis and synapse formation. So thus, I think what I really want to say is organoids are a nice model system, not just to study human brain development, but if you like to understand human specific disease mechanisms, they give you the sort of leverage or the system to go right from a mutation and altered epigenetic mechanism to looking at the altered neural development, perhaps then, which can give you insights into how the brain dysfunctions in some of these patients. And this can then be correlated with the clinical phenotypes which which is seen from the medical end of the disease. So I think that's it. I have only that much to tell you all about in the nervous system development, going all the way from mouse to the organoids. And if there are any questions, please feel free to ask them.
Bhavna, so what is the difference between interkinetic nuclear migration and mitotic somal translocation?
So one of the things is the movement itself. If you remember the movement in the ARGs, it's just a movement. But a mitotic somatic translocation is actually a jump. If you look at the movie, that it just jumps from wherever it is to the to an upper position. Now, the reasons why both of them happen are the same. I mean, it's thought to be, it's not yet known. The reasons is simply that to avoid molecular crowding at the zone of division in the apical radial glia and the INM, basically it is to give space close to the ventricular zone so that there's no molecular crowding and other progenitors can divide. Similarly, in case of ORGs as well, it is thought to, and plus facilitate the migration of the daughter cell. So it is thought to prevent molecular crowding. Now, very important difference is the players which are involved in this migration. In INM, it is your actin, microtubules, dynin, centrioles, all those are important for the migration, but none of it seem to be important for the MST. In fact, its MST is happens by non-microtubule like myosin, and the Rho-ROCK pathway is important for the MST. So these are some of the differences which people know till now, but I think more studies need to be done because the ORGs itself is such a new phenomenon. I mean, it's known for the last 10 years or maybe 10, 15 years. So, I mean, a lot of studies need to be done. For a long time, I think tissue scarcity was an issue, but now with organoids, I think people can do more manipulations to understand what particularly are the differences between INM and the MST.
Thank you.
Yeah, yeah.
Bhavana, I had a doubt. Where do these ORGs come from? Do they come from the basal, the apical RG cells?
Yes, yes. The ORGs come from the apical radial glial cells. Yes. The
And their cell division, are they the same? Do they both divide asymmetrically and form?
So I would put it this way. So the difference between ORG or a BRG, basal radial glia, and basal intermediate progenitor is that the ORGs undergo more divisions. They can undergo both symmetric and asymmetric division, which the basal intermediate progenitor does not. It has few restrictive, I mean, it only divides probably one time and then give rise, and maybe not even that, it just directly gives rise to neurons. Whereas the ORGs can undergo more number of divisions yeah, and that's the difference between in that subventricular zone. Between the ARGs and ORGs, they're actually quite similar in that they both can undergo symmetric and asymmetric division, but their markers or signature markers are different. Their movement of their nucleus is different. I think I'm not sure which probably one needs to look at is whether I mean, their capacity to generate the neurons is also very, very similar. But though people have not yet seen whether there are any differences in the output of the neurons which comes from these progenitors.
Okay, so like how we saw in the mouse development, how cortical neurons are formed from the RG cells. So even in the ARG and the BRG, do they both contribute to the cortical neuron formation?
Yes,
like
yes. Though yes, and though the formation of the upper layers, the superficial layers, is more from the ORGs. So the ORGs contribute immensely because the ORGs have expanded. They give rise to a whole lot of superficial layers, and the superficial layers are highly expanded in the humans. So it's all relative, right? Of course, the layer 5 and layer 6 have expanded, but the extent to which the superficial layers have expanded in the human cortex is much, much more than those. And the ORGs have been implicated to have contributed to those, to the expansion of the superficial layers.
Okay, thank you.
Hi, Bhavna. I have
Hi,
a
Ruchi.
experimental question. That is, so when in these stem cells, how does one distinguish a neural stem cell versus that of a radial glial cell? Because as far as I understand, both of these cells are known to give rise to neurons or astrocytes, right?
So what do you mean by a neural stem cell? Which neural stem? Because apical radial glia is also a neural stem cell for
okay, so it's
in a very broad term. Yeah.
Okay, so it's just terminology that's used interchangeably then. Okay,
yes,
okay.
Yes. It's all terminology. Yeah.
Okay, okay.
Yeah.
So Bhavana, I had another doubt. So I think when you had left the meeting, Sonia was referring to this experiment on using flash tag to kind of tag the neurons at different time scales. So could you?
That's the experiment which I showed to you in Tele et al. So I think while explaining, probably it got, yeah, maybe you should have told me before, but so this is the flash tag. Yeah. So what they, can you see my screen? Yes. So
yeah, we can see.
Yeah. So FT is flash tag. So the beauty of this experiment, and I think I'll explain this to you. And so what they've done is there is this molecule called flash tag, which only is taken up by these apical, you inject it into the ventricle, and these progenitors which are at close to the ventricle, they, the ARGs, only they take up this FT tag. And then what they've done cleverly is that they've let the ARGs take up these FT, and they have then harvested at 1 hour. So if you harvest at 1 hour, you will see that the ventricular zone progenitors take up the FT tag. So they are labeled. If you collect after 24 hours, you will see that the ventricular zone has taken up, but then they go on to form the subventricular basal intermediate progenitor or the newly postmitotic neuron. And if you would harvest at 96 hours, you will see now the neurons, differentiated neurons have now taken up this, this FT tag, right? So that's how they have now labeled the entire cortex across the radial axis using one molecule and harvesting at different time points. So you can easily fact sort these to take out the apical radial glia, to fact sort these to take out the basal progenitors and the newly postmitotic neurons, and fact sort these to take out the differentiated neurons. So this is the entire key of the experiment. This is the flash tag for the FT molecule which they've used.
So is the takeaway message from this that the VRG forms the ORG, and which then forms the postmitotic neurons?
Absolutely. So the take home message is a few, I think. I mean, I didn't want to bombard you with a lot of take home messages from this. What I really want you to take home 12 to E15 is the change. Can you hear me?
No, you got cut again. Could you repeat that?
Yeah. So yes, one of the take home messages across the radial axis is that the, and I think I showed it here. So, okay, I really didn't realize that it was this bad. Which means probably you didn't hear any. So can you see the take home message here? As you can see that as it goes from an AP to a neuron, can you see the signature just changing across here?
Yeah.
Right.
Yeah.
So the signature has just completely changed from a progenitor-like to a neuron-like. Yeah.
Yeah.
So that is one of the take home messages, that the signature completely changes, and the signature actually starts with more progenitor-like, and there is a differentiation, conserved differentiation pathway which the neuron takes.
Yeah. Okay.
Is that across the time, the cell progenitor itself goes from an introverted to an extroverted cortex. So which means genes like chromatin organization and cell cycle control are more in this progenitor. And as the progenitor becomes E15, more membrane receptors and control of membrane potential kind of genes come up. So which means now this progenitor is ready to take on the environment and sort of integrate inputs from the environment as it becomes a neuron.
Okay, yeah.
Yep.
Yeah.
So there are many take-home messages from this, but I just wanted to keep it very brief and simple. And I mean, you're right in the sense that if you characteristic signature, the basal has, so it goes from an AP-BP to newly neuron to a post-mitotic neuron. And each of them have very characteristic signatures.
Okay, yeah. Yeah,
Yep.
thank you.
Okay.
Yeah.
Yes. Yeah.
So here, introverted basically means that a lot of chromatin genes that are involved in chromatin regulation and cell cycle control are active at E12 compared to that of E15, where these genes are not as active as, let's say, membrane receptor.
Correct.
Okay.
Right.
And in terms of novelty of this study, so what I understand is what makes this novel is that they had used this FT injection.
Absolutely. Yes.
And
Because you could,
yeah.
right. Before this, you couldn't have anyways distinguished a ventricular from the subventricular progenitor. Right?
Okay.
You could fax purify it still, but
Yeah.
it's highly difficult because you're working with a highly mixed population. So the presence of this flash tag molecule was what made it possible, the fax sorting and very pure populations of, say, you could have never done a 1 hour harvest, because even if you do a 1 hour harvest, you will all end up you have to be absolutely sure, right? Because some of these markers of say apical, they are even expressed in SVZ. So then how do you then distinguish? So this presence of this flash tag has helped you completely segregate the two population and ask the question that what are the sort of unique markers between the two population, or perhaps markers which are shared between the two. So some of that is also part of the study, which I didn't tell you about. Like there is a lot of take-home message from this study, and I think more and more people will use this flash tag and now come up with their own questions.
Okay, so like just in terms of methodology, you label like it's sort of a lineage tracing, like this
Correct,
cell goes
absolutely.
there, and in the process you fax sort them and do a single cell RNA sequencing of the same starting cells.
Correct.
Okay,
yeah,
got it.
Sorry, I didn't realize that when I was saying all of that in the methodology, I think I was not heard properly.
Yes, your voice kept on going.
Glad you guys brought it out. Yeah, I have shared my slides with you, so maybe you guys can just go over it, and if required, we can have a discussion later on if one of you wants some more information on some of this.
Bhavana, I had a somewhat more general doubt. So in an adult brain, do we know if these RG cells and their fibers, do they still exist, or do they sort of degrade or recede, or is it only there in development?
Yeah, so the apical radial glia are all embryonic mechanisms. Though there are zones where the division continues. For instance, the DG niche in the hippocampus is one such area where the stem cells exist and they give rise to neurons throughout life. So is the, there is, I mean, there are a few other regions. I think it's called the, it's close to the lateral ventricle. So there are a couple of more places where the neurons are being produced throughout life. But those stem cells are not radial glia, and they're very different from the radial glia.
Okay. So these are only seen during embryonic development.
These are only seen during embryonic development. Yeah.
Okay.
So in terms of invertebrates and vertebrate brain development, it seems like only till so in invertebrates there's neuroblasts and some level of proliferation, but and that's the same thing that happens in vertebrates. But after that step or stage, both these systems are then become divergent. Like there's no
Yes.
parallel.
Yes. So some of the very starting mechanisms are highly conserved. For instance, I think the neural structures like neural tube, and maybe even some of the starting divisions of the neuroblasts, whether, for instance, the symmetric asymmetric divisions, they're very similar. I think Drosophila also uses the Par3 num mechanism to make asymmetric divisions. I think so. Though I must say that I've not done a very, probably there are a few reviews which discuss the differences between the two. But yes, some of these early mechanisms like division or the formation of the neuroblasts to the formation of the progenitors, they're very, very similar. But then if you look at the systems itself, right, like for instance, the neocortex, a structure like neocortex, maybe I talked to you all about the neocortex, which is a highly mammal specific structure. But perhaps if you go to some other systems like the retina, or I think though there, probably the mechanisms could be very, very similar. So what I really told you was a mechanism which is highly specific in the mammals, and going to humans, it's becoming more and more exclusive. Right? But if we look at retina, or for that matter, the spinal cord, I think the spinal cord is a very good system to study some of this, because some of the mechanisms of division in the spinal cord are conserved. For instance, the sonic hedgehog, or the interaction between sonic hedgehog and BMPs. Some of them are conserved. So as I said, there are conserved mechanisms on which your newer mechanisms have come up. And neocortex is one such example where some basic mechanism is conserved, but then on top of it, newer things have been added on to. And that's what I told you in my talk.
Yes. Okay.
Hi guys, can you hear me?
Yes, Sonia.
Okay. Sorry, who asked that question? I'm on my phone, so I can't see.
Yojit.
Yojit. So Yojit, can I ask you why you said that a lot of these, apart from division, all these mechanisms are different?
So what I gathered was, so like you said that, so in terms of differentiation, once you have these proliferatives, so the neuroblast or NSEs in mammals. So in mammals, like for instance, neurons are generated first, and then gliogenesis happens. However, in case of invertebrates or Drosophila specifically, the presence of neurons and the glial cells is happens at the same time. Correct?
Okay, so actually, yeah, I know Bhavna said that last time, but that's actually not true.
Okay.
So it depends on how you're looking at it. So I told you that there are in invertebrates, there are these two phases of neurogenesis, embryonic and post-embryonic. And then, so if you look within, so if you look at, just like invertebrates, you have the cortex, you have the retina, you have the spinal cord, and Bhavna spoke to you about cortical neurogenesis, but there are all of these other places where the rules, the general principles are the same, but the specifics are kind of different. Now, exactly like that in invertebrates, you take embryonic neurogenesis, and there is a temporal, if you look at individual neural stem cells, that not all neural stem cells generate glia in the embryonic phase, and the ones that do, there is a temporal distinction of when those will generate neurons and when they will generate glia. If you look in the post-embryonic divisions, in the type 2 neuroblasts, which are much more like the vertebrate division patterns, the INPs actually generate glia right at the end. So So there is temporal distinction of when glia are generated and when neurons are generated. What I didn't tell you about at all in invertebrate neurogenesis is how things happen in the peripheral nervous system. I spoke to you only about the central nervous system. And in the peripheral nervous system, the glia and neurons are generated simultaneously. There, the rules are, if you are not on, you become, I always forget, but I think you become neuron. And if you're not off, you become glia. So peripheral nervous system does things differently. Central nervous systems, there are distinct temporal windows where glia are born and neurons are born. Okay, so that was one of your reasons. You had other reasons, Yojit, to say what you did?
So the other reasons, no, I think these were like the first two main points that came to my mind when I said that.
Okay. The 2 points being, one was glia. What was the other point?
So glia and neurons, in terms of why they are divergent and why they are parallel, is like the proliferation of neuroblasts in invertebrates and NSEs in case of mammals.
So the proliferation potential is certainly different. And the jumping around of neural stem cells is certainly different. But otherwise, the division patterns, similar division patterns exist in both vertebrates and invertebrates. And molecularly, exactly what Bhavna said, all of your PARs and NUMs and inscrutable, in fact, as well, all of that is the same, where you need to have asymmetry generated via all those complicated molecules that I spoke to you about. Some that have been studied in vertebrates are the same, the PARs and NUM. And you need to align that to your cell division pattern, which inscrutable does in both vertebrates and invertebrates. So at the level of division as well, except for potential, there are similarities there as well.
Oh, now another similarity that came to my mind was how these gradients are formed. So even that more or less.
Very early patterning are very, very similar. Like I think some of your molecules, which are the culprits are the same. WNDs, BMPs, sonic hedgehog. And some of the divisions of, so as I said, there is one set conserved pattern on which then the extra things have come up. And particularly when you talk of a structure like neocortex, it has the add-ons have been a lot to the basic conserved mechanism. So the basic things like area patterning and all are very similar. Maybe the molecular players are not as similar, but the idea of having the organizer center or the gradient of genes, those are very, very similar between the two systems.
Yes, I think this should have been my first point of.
But no, but did you notice, Yojit, in the very, very early event of dorsoventral patterning, the things that set up the dorsoventral patterning in the very early in the vertebrate and invertebrate are the molecules are exactly the same. But what's absolutely fantastic is that they're completely inverted, because the nervous system is ventral in invertebrates, but it's dorsal in vertebrates, and it's the exact inversion of those molecules. When I first saw that, I was blown away.
Yeah, but yes, it's dorsal in the invertebrates, and later on, actually, the entire dorsoventral gives rise to the nervous system. It's just that it gives rise to different kinds of neurons. The dorsal gives excitatory, whereas the ventral gives the inhibitory neurons. So I think it's evolution in place, because as the functions evolved, I think some of these development events also evolved to include the new additional mechanisms, to make more or different kinds of neurons, more of the same or different kinds of neurons.
Okay, so can we ask you guys a question now? What are the rules? Maybe Varsha can tell us. What are the rules for assignments in the DevBio class?
So there are no rules per se, but so if you give the assignments, you can give them a deadline, and they would submit it on Moodle
Okay,
before the set deadline.
And have you guys had assignments from all of the modules that you have done?
Only Tina had given them an assignment, 2 assignments.
So may I ask what that assignment was, if you don't mind?
Yeah, sure. So she had asked to write the question behind the experiments in a paper, in an old Drosophila paper.
Particular. Okay, okay, got it. And have you guys had any other assignments apart from that? Varsha, have you had any other assignments apart from that?
No, just those, those 2 assignments. So there was one more assignment which was the same thing. There was an experiment, and she asked to describe the question behind the experiment.
Okay, so maybe what Bhavna and I will do, we'll sit together today and we'll plot an assignment. Do people prefer certain kinds of assignments? Do they prefer like an MCQ or a write-up, or is there something like that that we can give that will cover both our sections?
Oh, I mean, yeah, either of them is fine, I think, because until now, in NCBs, I think people only give like descriptive type answers as assignments.
Okay, so Bhavna and I will mull over this and give you an assignment, and then Varsha, maybe we'll talk to you about what we should set as a deadline. I know you guys are doing lots of courses right now, so we don't want to burden you with a very tight deadline, but we'll
so
talk
usually
with
it's a week, Sonia.
Okay.
So if you give them time
to finish the assignment, is that what you're saying?
Yes, a week to finish the assignment. Yeah.
Okay. Yeah. And then, sorry, maybe we should. It will be evaluated, right? Or it's just
yes. You can evaluate it and give me your comments and grades, both of which I'll update on Moodle.
Yeah. Okay.
Okay, great. So then, do people have any more questions for Bhavna or me before we end this? Bhavna, did you want to say anything?
No. I think if people have questions, we can take it up. Otherwise, then we will then come back to you with an assignment.
Okay, what is the thing they do at auction? Someone had helped me last time. Going once, going twice. Gone for the question time. But no, honestly, please, if you have questions, do shoot us an email. You have both our email addresses, and you'll hear from us on assignments. Okay, thanks a lot, guys. It was really fun and really nice getting all of these questions from you all. I hope